So hello everyone, Oliver here. Hi, welcome again to another Saturday live stream, microscopy live stream. That's the specimen I'll be putting under the microscope today. It's an onion. It's a classic traditional specimen. Yeah, and I'll be trying to do a little bit of staining, a little bit of plasmolysis, some plasmolysis experiments, and so on and so on. So we're gonna again see how far we're able to go today. Yeah, it's the first specimen normally that you put under the microscope when you start off with microscopy, but it's I think also a very maybe even underrated specimen. It's pretty interesting, I think, if you know what to look out for, and that's what I would like to do today. Of course, I also would like to pick up some of the things that I started last week, specifically this little tube here that I prepared. Look, I added some hydrogen peroxide to this water sample. Something quite surprising happened here. I put it under the microscope just a few minutes ago, and the hydrogen peroxide was not able to kill the life in this tube here, and it's actually started to grow bacteria as well. Maybe if we have time, we're going to have a short look at this as well. So, like every week, hello around the world, because it does take a couple of minutes for people to join in. There's usually around a 30-second to a minute time delay also between essentially when I say something and when you're able to see it. It's because the video is buffered, so I'm not able to respond always directly, but greetings from Ireland, Vietnam, Florida, United States, Vietnam, okay, from Greece, Northern Germany, Upper Austria, Georgia, Northern France, Netherlands, written in Esperanto even, from England as always, North Florida, Huntsville, Alabama, and so on. So that's basically the thing. So what I would like to do is the following. What we've got here, this here is of course an onion. It's a red onion, as you can see, and the reason it's red is because we need the pigment. And what I want to try to show you today is just some regular onion cells under the microscope. It's pretty standard stuff. And then I would like to also show you how you can prepare some of the red cells of the onion for microscopy. And then I'll be adding a little bit of salt water to observe so-called plasmolysis. And then also a little bit of staining of onion cell nuclei. Now, the success always depends a little bit on how fresh the onion is. There's always a little bit of trial and error going on. Sometimes it works well, sometimes it doesn't. But that's the whole point of simply trying out new things. Now, last week I already asked you that if you have any suggestions on things that you want to see or that you want me to put under the microscope, please feel free to write comments, and some people did. Of course, I'm going to pick up on that as well. Okay, and I've seen that there's again a hello from Sri Lanka. Okay, and what I'm going to do is I'm going to go to live chat, so sometimes the sequence is a little bit different here. Okay. So that's kind of the idea, and we're just going to start off. This is, by the way, of course, one of those the first preparation steps that you do. If you want to put anything under the microscope, then onions are basically the first ones to try because they're so simple to prepare. All you do is you make a cut, like this here, and you basically. And I've done this already so many times before, so maybe many of you are already familiar with that. When you put it out, then you can actually see, of course, the different parts here. And usually what I do is I use a clean knife, of course, because the onion being food, I do not want to throw away, right? And so what you see now here are the layers. Okay, and I like this a lot because you can actually, if you've got a good microscope, then you're also able to see the movement inside the cells of the onion, and those cells are fairly large. Then you have to basically do the following. You have to find between the layers here, you have to find this. I dropped it. This is what you call the onion skin, and you can try to carefully peel it off. You see it's basically a thin layer, and usually it's around a layer or 2 layers thick. And what I usually do is I simply take it and put it directly on a microscope slide without any water, because I'll be adding the water in later. The reason is because this way it's easier to spread. Okay, and then I directly drop a cover glass on top. Yeah. By the way, if you're new to this format, please feel free to ask questions, off-topic questions, which are still related to microscopy. I'm going to try to answer up after approximately 1 hour. And it's also it's customary when if you have, I don't know, comments or questions to other members of the chat, then please feel free to do this as well. But if you basically have specific questions to me, then please indicate this with @Oliver or @Microbehunter. Okay. Yeah, there's already a comment from Ever Smaller. Maybe in a future stream, extract the pigment using alcohol or by stepping in water and then use it to stain something with a different pH and then mount the specimen. Yeah, that is, first of all, probably alcohol for extracting this pigment might not be necessary because, to my knowledge, these pigments are known as anthocyanins. Or anthocyanin, and basically they're water soluble. So it might not be necessary to use alcohol. I could try that. Maybe it's easier this way. The question that I have, and that's going to be a little bit the trick maybe, is how can you concentrate it to the extent that you can actually also use it for staining. And the interesting thing about these pigments here, also in red beet and other red vegetables, is that the pigment is pH sensitive. So this means that it's able to change color depending on pH, and that's why there is essentially the comment here. Yeah, so that's basically the onion skin. I'm going to drop this cover glass directly on top here. Okay, and only now am I going to add some water. Okay, so I need my pipette over here. And usually what I do is the following. I'm just going to take this jar here, add a little bit of water. And the reason why I add the water only now is because it's simply easier this way for me, because if I add it first, then there is, it starts to roll up maybe, okay? So I'll be adding simply a little bit of water here on the side, and you can, you can actually see how it slowly starts to seep in. So let's, now here we go. Yeah. Now, will there be air bubbles? Yes. So that's actually with those flat onion skins, it's pretty common that air bubbles are in there. If there are not too many of them, then yeah, it'll still be fine. Yeah, and because normally I'll simply try to find a part here which basically is more or less free of air bubbles. Okay, so I'm going to give it now a try again, and I'm going to switch over to the microscope view. And one of the interesting things that I realized is that even though while the cameras actually do work, it takes significantly longer now to switch. I don't know why this is the case. Yeah, so I hope that it's not acting up, and this is how it looks like, and I think it looks pretty nice. Again, people keep on asking me a little bit about the color, because essentially there's this nice bluish background color. This has to do something a little bit with the microscopic optics that I'm using. It's called DIC, differential interference contrast microscopy, and this gives it a lot of color. That's one of the reasons why I bought this microscope, because for YouTubes and live streams, it's nice to have some colors. So you're not going to see it this way. So what I'm going to do is I'm just going to flip away the filter, simply to give you a more authentic look, and that's basically how you are probably also going to see it under the microscope. Okay, yeah. So yeah, this is basically with the DIC. It uses polarized light, and then you see you can do all of these color changes here, and it looks kind of pretty and everything. Yeah. Sure, but these are not the natural colors, right? So if you want to see it in a normal way, and later on I'll be using that when I'm doing the staining, it looks basically like this, right? And what I'm going to be doing now a little bit is I hope that I'm able to see that, what I want to show you. I'm just going to zoom in more and more and more, and you can actually see that, and I actually do have an arrow. Yeah, these are the cells, and all those round things that you see here, that's the nucleus, right? And that basically that contains the DNA, and here on the outside you have a thick cell wall, right? So, and I'll be zooming in now, increasing the magnification now to 40 times. I always have to go up with the brightness a little bit, right? And then let's have a look here, a little bit at one of those onion nuclei. And I found out it depends a little bit on the age of the onion as well, whether you're able to see this properly or not. So let's go up all the way to 60 times. Okay, it's still bright field, right? And what you're able to see occasionally better than sometimes better than in other cases, and yeah, I'm just going to look around. This is actually that there is a little bit of movement inside the cell, and what I generally recommend is that you try it in time lapse. So you make a video of, let's say, 10 minutes or so, and then you play it quickly, and then you can actually see the movement of those tiny dots inside. So I'm going to just find a, you know what, I'm going to, yeah. Could we try to stain organelles? I'm going to, this is a question. I would like to show you a very simple staining technique to stain the nuclei. Okay. I know that there are many more cell organelles inside a cell. You actually have to use, yeah, ideally some stains that are highly specific, but it's possible to stain the nucleus as well using regular ink. That's something I would like to show you. So yeah, let's have a look here. Okay. Honestly, I've seen already some specimens where this actually is better visible. But occasionally what you're able to see is that those tiny little dots that you see in here, you can actually see that there are more here on the side. Yeah. Sometimes in an onion cell you see a central region which does not contain these dots. And those dots here are in many cases so-called vesicles, transporting substances, and inside the onion cell you have this large vacuole where essentially you don't have those tiny vesicles, and if you actually observe it now for a longer time period, you might be actually able to see that they're moving around. And now several months ago I did make videos about this, about the time lapse, and it looks quite interesting to actually see them moving around, streaming around, right? But you'd have to actually really go up with the magnification quite highly to actually see them move. Yeah, that's of course here, that is the nucleus from before. Yeah, and you just look around, and then sometimes you are able to see, yeah, see it a little bit better than in other times. Yeah. So let me see. Yeah, so I'm going to show you another onion preparation techniques of cells as well. So how we can actually see the red cells of the onion, right? Yeah. So that is essentially, yeah, if you focus now, you can actually now see also the cell wall. Okay, now a little bit of biology maybe. Plant cells have a cell wall made of cellulose that's fairly thick and visible over here, and very thin on the inside, and so thin that the microscope is not able to resolve it. You have the cell membrane. Okay, that's I think I already talked about this as well. When the early microscopes were not really very good, when people put plant cells under the microscope, what they were able to see were mostly the cell walls, right? And they could not see so much of the internal details. That's why they needed to actually use specific staining to make the parts visible. But apparently what I read in some of the historic accounts is that at the very beginning, the people were very, very interested in the cell wall. And as a matter of fact, the people at that time were actually debating a little bit. Yeah, the plants are made of cells, but what is actually alive? They actually thought that, not thought, but this was actually one of the ideas, that they thought that essentially living things like plants, yeah, they thought that maybe it could be, maybe the cell wall is the living part, right? And maybe these are just empty spaces in the center, right? Because they could not see the cell's contents. So they thought that maybe living things are kind of like a sponge, right? And so maybe the cell wall is the living part, and then you're just basically, the central part here is just empty, right? Of course, now we know that it's actually exactly the other way around, that the cell wall made of cellulose is actually outside of the cell and considered to be non-living, right? And the cytoplasm on the inside is actually the living part. Yeah? So yeah, so I'm just reading some of the comments here. Could we try to stain organelles someday? I'm going to try to stain a nucleus today, yeah, using very simple staining, yeah, so that you can also try it at home. And in the area without dots, so why in other specimens we see chloroplasts move to the outside? Yeah, that's correct. Look, an onion like this, of course, does not do any photosynthesis. It's not green. It's under the ground. There's no light. And generally what we have in a plant cell is we have the cell wall on the outside, like here. Then we have a very thin cell membrane, which we're not able to see because it's so thin. And then what we have is a so-called a large central vacuole, which is filled with water and minerals. And the cytoplasm, the living part, so to say, is kind of compressed on the outside. And this is also where we often find the chloroplasts. So, yeah. Non-living, I don't understand that. It's all living. It's a little bit of, how do you say, a question of definition. I think it's an interesting definition. The thing is the following. And I understand it's a little bit of a question of debate and definition, so it's more of a question of wording. But according to the cell theory, if you see things moving around inside the cell, like chloroplasts, or because of so-called cytoplasmic streaming, you see stuff moving around inside a cell, then those things that move around inside the cell, per definition, are not considered to be living. Now that's a little bit of a weird thing, I get it, because per definition only the whole cell is alive. I know that this is maybe more kind of a language thing, but in order for something to be alive, you need to have at least one full cell, right? And actually, what is now the border of the cell? Yeah, the border of the cell is the cell membrane, and everything that's outside of the cell membrane is basically considered to be outside of the cell and non-living. I know this is kind of a little bit of a debate, right? Where do you want to draw the line? So I do understand that if it's a little bit like asking, I mean, what about hair? Hair, strictly speaking, is non-living. It's not made of cells, right? But it's produced by our skin cells, and it's protein that's kind of given off to the outside. Yeah, so hair is not living, but it's actually made by living cells, right? So I totally understand it's a little bit of a question of debate, maybe. Yeah. So I'm going to. Could we try to stain organelles other than like a nucleus, like mitochondria, endoplasmic reticulum, and so on? And the thing is, that is a little bit difficult, and because I want to explain this a little bit. There are certain staining methods, like I think certain forms of silver staining, where you can actually stain the Golgi body. The Golgi. I think it was actually Camillo Golgi who discovered this or developed the staining method. That's why the Golgi body is named according to him. But you see, many substances inside a cell are stained in a very unspecific way. So if you really, really, really want to have highly specific staining, what you need, unfortunately, is you need fluorescent microscopy and specific antibodies. Yeah, and those antibodies, they will bind to the structure that you want to stain, and then this structure starts to light up. Yeah, so that's essentially highly specific staining, which is used, of course, in research. But I would say traditional staining methods are probably going to be a little bit difficult. DNA staining works. Okay. Yeah. So. It's the dead particles that the cell use for the cell wall. Well, I think what you mean is the following. What is the cell wall made of? The cell wall is made of a cellulose, and cellulose are long chains of glucose, which is a sugar. And when you connect it together, then you get cellulose, right? For example, paper is made also mostly of cellulose, or cotton is made mostly of cellulose, right? I'm a biology teacher, and I usually, when I do biochemistry with my students, I kind of surprise my students a little bit by saying, well, the underwear that we wear is made of cotton, is essentially therefore made of cellulose, is therefore made of glucose. So the students want to know is, well, if it's made of glucose, it's a sugar, why can we not eat it, right? And because we do not have the enzymes to break down the cellulose, we're not able to digest it. Okay, so but I digress a little bit. But maybe for some of you some time ago asked, I had some biology questions as well, so I'm also willing to address it. I personally, what I have to say is the following: is that those cells of an onion are quite large. If you actually hold the onion skin up against the light, you might be actually able to see them in a very small way. Definitely, you are also able to see it with a strong magnifying glass. So if you I've got one over here, right? Yeah, if you hold it, against your eye, and then you hold, yeah, you might actually be able to see the cells as well. Yeah, you can, of course, also, I remember a long time ago when we still had those overhead projectors, so where we had those transparency foils and project in a classroom, yeah, and if you put an onion on there, an onion skin on there, you could actually see the cells also projected on the wall. Yeah? So that's essentially, yeah, I will quickly go through some of the comments here. Okay. I've been using very strong laser diodes. I can use the lens to create a highly visible sheet of light, and was wondering if you've ever used such beams for light microscopy. I think I've not used that, but you know, is it a little bit like this, that maybe you take what I heard? I have not tried it yet. Is it for if you take one of those laser diodes and when you shine it on a let's say onion skin or something, then the light is kind of spreads apart and you're getting actually a projected image on the wall. Yeah, I've not tried it, but I heard that something like this might actually be possible. Huh? So yeah, quickly look if there's anything. Yeah, Golgi body can also be stained with neutral red. Okay, I just wonder if the Golgi bodies are not a little bit too small to see. Okay, but we're going to find out, right? So that's basically how it looks like. I'm just for comparison, this is bright field, right? So this here is this one is DIC. Now we can actually see the some of those yeah, tiny vesicles much better. Maybe I'm just going to go up a little bit with a how do you say, with the magnification. Maybe we are able to see the movement a bit better. Let's go up yet further. Yeah. And here we are able, let's turn this, let's make all of the necessary adjustments. So, and actually then we are able to see a little bit more that you see that it seems to be fairly smooth on the inside. This is where the vacuole is, and on the outside here you see those tiny little dots. And if you were to now make a time-lapse video also over here, yeah, then you could actually see them move. Yeah. Another thing that you can occasionally see, maybe not here so much, but occasionally it's also possible to see how the cell walls actually have gaps in between them, where basically the cytoplasm is connected, the adjacent cytoplasm is connected. Yeah. Yeah, it's called plasmodesmata, it's called, right? And this is also one of the ways how the cells are able to communicate with each other. Right, yeah, but you can see that indeed very often on the outside towards the cell wall, this is where you see yeah, a lot of the cytoplasm. Yeah, so this is simply just yeah. So what I'm going to do here again, a nucleus. Yeah, maybe let's have a look. Yeah, sometimes like this. Okay, you know what I'm going to do just for the sake of comparison. So this is a DIC. And I also have a 40 times phase contrast. And let's try this. Let's open it here a little bit. Yeah. And that would be now phase contrast microscopy. Phase contrast is also a specific technique, usually very suitable for bacteria and other very thin specimens. And what it does is that it converts transparent, non-stained specimens into different darkness. Okay, so you get almost something like a black-white image, which does not always look very nice, maybe, because it does lack a little bit color, but sometimes you're able to see things that you were not able to see before. Okay, so there is Simply Electronics is now clarifying this a little bit. Actually, my idea with a high-powered flat laser beam is to illuminate a specimen from the side. Oh, I get it. I could imagine this might create some interesting 3D lighting or enhanced details. Yeah, that is indeed possible. That would be a little bit like so-called oblique illumination. And yeah, this is indeed something that is possible. Generally, there's always the problem, if you look up here, if you want to have a light coming from the side, yeah, then usually sometimes the objective casts a shadow, and then it's difficult to actually get the specimen illuminated. So I've some people sent me already some pictures of where they try to arrange lasers, very tiny laser, very flat even, or you can try to do some kind of, not lasers, LEDs. Right? So there are indeed several possibilities, which I have not all tried out yet. Okay. So I found a ladybug today that looked perfectly fine, but was dead. No obvious external damage. I assume age. Okay, now it's in a tube of alcohol. Exactly. Also put it into alcohol because you want to kill bacteria, of course, and to prevent decomposition. Okay. Okay. Yeah, I noticed that your chloroplast videos usually have chloroplasts on the outside edges and not in the center. Yeah, indeed, because in a plant cell, what you have is in the center is very often occupied by the central large central vacuole, and you've got the cytoplasm and the cell organelles compressed to the side. By the way, that's how plant cells actually are stable, right? Because there's this pressure that's built up. I just see that I'm doing a little bit almost like a biology lesson now. Not that I mind. Okay, the wrong filter. Okay, so but what I would like to do now is I would like to actually move away a little bit from this standard bright field microscopy view, right? That's basically the onion, the skin. I would like to now show you how you can actually prepare the red onion cells, because that preparation technique is a little bit different. Okay, so I need to switch back again to the desk view. Okay, so here we are again, and I'm just going to grab this one over here, right? And you can see that this red layer of pigmented cells cannot really be peeled off very easily. So I'm going to show you a little trick how you do that. You take a knife, but the knife should not be too sharp. It's really important, yeah. So what you do is you just basically cut right in the middle here. It doesn't even have to be very thin, right? You just kind of cut into the how do you say, into the, the flesh or the bulky onion here. And then you use your thumb and you really press hard against this layer of cells. And then you got to be really brave, because what you want to do with trial and error, you want to kind of rip off the top surface. Okay, actually, I got this quite nicely done. And then you try, and you see it's very thin. And if you're lucky, then many of these cells are intact. Okay, sometimes, I mean, actually, this is actually quite nice because the whole thing came off. Yeah, it's actually normally not the case. And then what you do is as before, you put it on here, on a microscope slide. And you have to, of course, there's this thick part still here, which I'm just gonna cut off. Because it's got to be thin. I do have a scissors somewhere. Here it is. You might also want to use a sharp yeah, you know what? Scissors are so boring. Scissors are so boring. I'm just going to show you one of those. This here, as we deep printed this here, actually for this is supposed to be a microtome knife. Yeah, but I'm just going to use it here as well to simply cut off. I wonder if this works. Yeah, here we go. Yeah, I was able to cut this off. Some tweezers. So I'm just basically removing the thick part, right? Yeah. And now again a cover glass. Okay, and I'm not going to be adding any water. No water added. Just a little bit of And so this is almost like a dry mounted specimen. The reason why I'm not going to be adding any water now is because I'll be adding some salt water in a second, and we're going to see what actually happens, right? So I'm going to now switch back to the microscope view. Again, it's kind of funny because it's now taking so much longer to actually switch. Yeah. As an engineer, I'm really curious about the effects of different light on microscopy, UV light, infrared, thermal imaging, who knows? Yeah, many of these things actually do exist already. Yeah. So, especially on the university level where microscopes are so modular, what they're doing is they're doing all sorts of different trials here. So, and that's basically how it now looks like under the microscope. It's, without, we're just going to look at a place where it's kind of more or less homogeneous, and you can actually see that most of the cells here are actually red. Well, obviously. Let's have a look at some of the cells here on the side, and you can actually see it. Look, some cells seem to be white, and the reason is that those cells that are white, very easily visible here, they have broken open as I ripped it off, and the cell contents has flown out, so they're kind of damaged. right? Depending on how well you're able to do that, yeah, many cells can be damaged like this, and you have to just find a few cells that are essentially still intact, right? So this is essentially what we, yeah, here on the side, for example, the more, yeah, that's one. See? That's actually quite nice. You can actually see very nicely that a couple of these cells are still intact, and those white cells over here are still broken. Yeah, have ripped open, and the cell's contents has been lost. Oh, I bumped it. Yeah. So what I would like to do now is I'm going to now add a little bit of salt water. And one has to be careful because I do not want to make the objective wet. I'm just going to go up a little bit now with the magnification as well. Okay, so I have to quickly remove some of the pop-up windows here. Yeah, so, I'll be now adding a little bit of salt water to the edge of the cover glass, and I'll allow now capillary action to do the rest. So basically the salt water will be pulled in. I'm just looking for my pipette tips, which are misplaced. Oh, here they are. Yeah, there's so much mess on my table that I have problems finding those. So, and look what I got here. This here is salt water. Okay. You can, I don't know, it's nothing special, just regular tap water, and on the bottom you can see salt. I warmed it up in the microwave a little bit to speed it up, the dissolving, but it's saturated. It doesn't even have to be saturated. Yeah, just regular table salt. And look, I don't feel like switching around. I just take up a small drop here, right? I'm now going to put it on the edge here of the cover glass, and I'm going to allow the capillary action to pull it in. So you should actually be seeing how the cells of this onion here are essentially submerged in the salt water. So we're ready to go now, okay? And look what happens. Okay, so I'm going to carefully. Okay. Not yet. I have to probably add more. It's slowly moving. Let's see. Yeah, it's working already. I need to explain. So, yeah, let's move a little bit to the side here. Here we go. Okay. Yeah. So, yeah, let's maybe also from the other side. It's not quite fully covered yet. I'm kind of worried a little bit that I got some of the salt water on top of the cover glass, but it's already working. You know. Up, here we go. Yes, very nice. Up. Ooh, what's going on here? Okay, you actually see what happened here? Let me quickly check if there is good. The objective is still dry, okay? And look what happened. Here is the following. You see that some of the cells over here seem to be completely not affected. These cells are kind of damaged. And the other cells here, what happened is that all of a sudden those white areas started to appear, right? And this basically means, and the central part, the one that's pigmented, became darker. So what's going on? This is called plasmolysis. And in just a second or so, we're going to reverse this, right? And what happened? Let me see if I can actually go up with the magnification without actually touching the objective. Let me see. No, I have to do the following. I got a little bit of water. On the surface. Did I? Yeah. Okay, so what I'll be doing, look, I'm going to go to the desk view again, okay, and I got some water on the surface. The objective did not touch it, luckily, but I have to remove this liquid, otherwise I'm not able to go up with the magnification further, right? So that's fine. I'm just going to dry it as well. Okay, it's still submerged. In salt water, so that's going to be fine. But again, I do not want to have any liquid on top of the cover glass. Okay, so this should be fine. Let's have a look. Okay, I need to change over again to the microscope view. plasmolysis, yeah, it's called. It takes, yeah, here we go. I need to, and usually this is, by the way, this is a standard experiment that I'm doing with students, because it does tell you quite a bit about the nature of cells. Okay, so I'm going to use this here as an explanation here. So I'm using now my 40 times objective, and what you see over here, this is when you add salt water, the actual size of the cell wall, did not change. It's still here on the outside. Yeah? So that nothing happened here. Yeah? The cells actually still have the same size, but something interesting happened inside. On the inside, you see those white areas appearing, and those white areas are places where the cell membrane separated from the cell wall, because the contents of the cell started to shrink. Because of the salt water, water moved out by osmosis, and the contents started to shrink, and as interestingly, therefore, the cell membrane started to separate from the wall. It is an analogy that you can use, like for example, a balloon that you blow up inside a box, right? And if essentially, yeah, there's not enough air in the balloon, then it's going to be very soft and it's going to separate, right? So that's basically what we're seeing here. So what we have over here, and that's too thin to see, but right here on the edge, this is where we actually have the cell membrane, which originally was connected to the cell wall. And so that's one of the things that we see here. And there's another interesting thing that occasionally you might be able to see, is that basically it's still connected to the cell wall at certain places. It's not completely separating. So we're just going to look around here a little bit. And the reason is because these places are places where the cells are connected with each other. These the so-called plasmodesmata that I talked about before. Yes, you see some of the cells here do not seem to be so affected. So either the salt did not reach those cells yet, and or they were damaged a little bit. But yeah. Yeah, so you can actually see. Let's find some places here. Yeah. Yeah. Here we go. Yeah. You can actually see how thin then sometimes yeah, everything becomes here. And if you actually look at this very fine place here, then you can actually see it's still, some of these parts here are still connected to the cell wall. Yeah. Which shows that the cell membrane is not yeah, completely separating. Yeah? And that is actually, yeah, the cell membranes are quite elastic, yes. Yeah? So the salt solution concentrates the color pigment. That's one of the questions. Well, indirectly, yes. What happens is it becomes darker because the water moves out. Evidently the pigment was not able to move out. It stayed inside the cell, and therefore the inside of the cell got darker. Okay, so that's the, yeah, so that's kind of the yeah. The cell membrane is elastic. You can actually imagine the cell membrane is also very fragile. Okay, you can a little bit imagine a cell membrane like a soap bubble. Actually, chemically, it's quite similar even to soap bubbles. So you have, for example, two soap bubbles. You can join them together, and then it becomes larger, right? As a matter of fact, the cell membranes are also able to do that. And don't worry, I'm going to try to reverse the whole thing by adding some regular tap water again. Okay, yeah. So that's let's go a little bit down with the magnification again, just to show this to you. Yeah, that's basically how now it looks like, right? Yeah, and you can actually see again that some of the cells they kind of remain a little bit brighter pink, and this could be indeed a sign that those cells maybe had a little hole, a gap, and therefore, yeah, salt water was maybe able to enter. This is people, that's the reason why you should not be drinking salt water. Yeah. Why it's so dangerous, because what happens actually is if you drink salt water is that the body loses more water. The cells actually lose more water. Yeah? And this whole process is of course called osmosis. Yeah? So that's the thing. Yeah. So what I would like to do now is I'm going to try to reverse this whole process. by adding simply a little bit of tap water. But this, of course, only works if I remove the salt water first. Okay, so I'll be doing that. Let's go back to the desk view. So I'm going to take this. You can even you can even see how the salt started to crystallize a little bit. So I'm going to carefully remove this thing here. I'm just going to, you know what? Just going to place it on here. You can actually use some how do you say? You know what? I'm just going to use a fresh slide here, right? Because I don't want to have any salt on here. Because I would like to reverse this, I'm going to remove also any excess salt water that I have on here. Of course, I want to make sure that everything remains nice and flat. Yeah. Because if there is too much salt present, then it's still going to be too concentrated even when I add tap water. Yeah. So it's still kind of flat. Okay. That's also possible. The recommendation, add tap water from the side and then using filter paper or tissue paper to remove the other water from the other side. I have been doing this as well. Yeah, so that actually also does work. Yeah, so thank you for the suggestion. So we're basically here like before, with the difference that everything still seems to be, let's see until it switches on again. Yeah, here we are again, okay? Okay, and now what I would like to do is I would like to see the reversal of this process by adding a little bit of I don't know, maybe this is nice. Let's make it a little bit bright. By the way, that's not all bright field, okay? So, and the arrow, I want to get away. Let's add some regular water. So I have to remove the tip, and I would just going to add some, just like before, I've got some tap water here. And let's do the same thing again. The focus changes. Let's quickly refocus, and let's see if it's able to start filling up again. I don't know if it has actually touched it already. Yeah, here we go. I think it's slowly starting to change shape. And then maybe we're also lucky to see some of the cells pop on the inside. Yep, I have to refocus because, of course, every time when I add water, then what happened here? Here we go. Yep. See how the water kind of now touches the cells, and the cell's content starts to become a little bit rounder. And I'm going to add a little bit more and okay, you see how the water starts to move forward. How does the pigment show in phase contrast? I can try this as well. Okay, but where is this? Let's see. It could be still a little bit too much. Here we could actually still be a little bit too salty, maybe. So I'm looking for a place, or I'm going to go down with the magnification to see if there are certain areas. Here, for example, you can actually see how the cell's contents now has start to become a little bit rounder, as it starts to fill up again. Okay. And occasionally, look over here, what happened here, it's all pink. I will tell you why. Because some of the cells start to pop on the inside, and sometimes we're also able to see that. Yeah. So I'm going to add still a little bit more water. Okay. Because of the salt in the center. Yeah. Okay, those cells here seem to be quite round. Okay, you can see that they slowly started to fill up again. Yeah. So essentially, what I'm doing right now is I'm kind of reversing the process, and occasionally you might actually see them that they start to pop on the inside, and then everything starts to become evenly pink. Yeah. So there's another question here. Are you getting a greater concentration of diatoms in the plant from last week from plant digested with the hydrogen peroxide? That's where I discovered something quite surprising, actually. Yeah. So yeah. Did you actually now see what's going on here, right? Yeah, you see that it's slowly starting to spread. So this the cell membrane on the inside seemed to have popped here, and then the pigment starts to spread. Yeah, and you start to kind of fill up the inside of the cell again with the pigment. Yeah. So you see that that's actually quite a nice thing that I highly recommend that you play around with a little bit. Yeah. So this is, I even in the past made a couple of videos here, but you see that if you just look around here, you can see, look, all of those cells here on the side here, yeah, they start to become uniform pink. And the reason is this is where time lapse would help. Yes, definitely. Yeah. You can actually see how then the cells start to, so maybe if we're a little bit patient, maybe we can actually see also some of those cells pop open again. Yeah? You can also see over here on the inside. Here you can see how the cell membrane has separated now, and on the inside you have the nucleus. So this is quite a nice, how do you say, little demonstration Is this because you added more water than you took out? I think it's not the amount of water that's so important. This is a question of concentration, of salt concentration, right? And what I've done is I've then removed the salt water completely and added tap water to push salt concentration down again, and I was able to reverse the whole process. Yeah, but of course there is still quite a bit of salt left, and for this reason, it's depending on the region where there's more salt under the cover glass. On some places it's happening faster than on other places. Yeah, but I think you do see quite nicely that it's starting to slowly fill up again, and that the contents, which was kind of very irregular at the beginning, now starting to become rounder and rounder. And in time lapse, you could actually see how it starts to expand more and more until it pops on the inside, and then the color becomes more uniform. Yeah. So you see over here as well. So look here. Maybe if we're a little bit patient, maybe we can actually see that how this one over here also will start, if we're lucky. Yeah, maybe it's gonna pop open as well, and then the dark pigmented color is gonna start spreading inside the cell. Here maybe it happened. Yeah. If you wash the sample in water and then put it back on the slide, would the rehydration happen faster? I probably would think so. The reason is because because then you're quickly removing any salt that was present. Yeah. If you just use a larger sample of water, then but then you would not be able to observe the process under the microscope. The point for me doing it directly on the slide is simply so that we can actually see the whole process in real time. If I were to take the whole onion piece and put it into salt water and in tap water and so on, I would not be able to watch this process, which is actually quite fast. Yeah, but I want to be able to follow it under the microscope. Yeah. So I'm just checking the time a little bit. So what I'm going to do, we're just going to, maybe go down with the magnification yet further. And then you actually see how this actually looks like now. Yeah, so evidently over here, the water was not able to reach it quite yet. Here as well. Let's go totally down with the magnification. That's basically how it looks like. You know what I could try? I mean, that would be a possibility. Let's try that. Let's remove the water again. Look, you can actually see how there is a look. now the pigment started to kind of run out. So I'm going to quickly clean this off. Carefully maybe remove any water. So, and let's have a look, yeah, one more time. Scope view. Yeah, it takes some time again for it to come back. So this is how it looks like. Ah yeah, there's still some moisture left. Let's add a little bit more maybe. And let's see if, did you actually watch some of them pop? Let me quickly zoom in. Pop, yes, this one here on top. You could probably already see it happening here, right? Yeah, it starts to expand a little bit and then it pops. Yeah, and then it's actually quite nice to be seen here. Let's add a little bit more water. Maybe we're also able to see those pop. Okay, a little bit more water. It does take it does take a little bit of patience to observe. Yeah, and maybe if you missed it later on, this video will be, of course, visible online again, so you can kind of rewatch everything in case you were not able to catch everything. But let's. This seems to be a good area, so let's add a little bit more water. And maybe we're able to see. Patience is all that's needed. Yeah. Yeah, one of the things that I kind of find surprising is, look, over here. Yep, did you see this here? It popped here, and then over here the content started to spill out. One of the things that I kind of find surprising, and I do not have a full explanation of this, is that why it is not able to expand to the full size again that we had at the beginning. This is something I don't fully understand. It was able to shrink, sure. And then when you reverse the whole process, then it starts to expand again a little bit, but it pops even before it reaches the maximum size. That's something I don't fully yet understand. Yeah? But you can see that this one over here quickly popped, and now the red pigment started to spread again inside the cell. Yeah? So let's see if we were able to see something similar over here in the others as well. I don't know. We're going to see. Yeah. Yep. Did you see this one over here? This one here on top popped, and now yeah, it's also going to start spreading. I don't know. I think you might have to check in this channel. It was several years ago, maybe 3, 4, 5 years ago. I made actually a video with time lapse where I basically showed this to you as well. Yeah. There's this gigantic air bubble up here, so maybe we can still add a little bit of more water. I mean, you get the idea, okay? Yeah. So here we go. Yeah. By the way, that's also one of the reasons why it's not good to drink distilled water, right? Because our body cells would actually also experience something similar. And by the way, if you want to try some similar experiments with red blood cells, it's really nicely visible with red blood cells. Okay, so the red blood cells, they really change their shape quite a bit if you add salt water. Yeah. So, yeah, just wanted to show this to you. I'm checking the time a little bit. It's almost now 1 hour. We'd have to be a little bit more patient to see the others. Yeah, pop, but I think those few that we've observed, you might have been able to see it as well. What I want to show you now very quickly is a very simple staining technique for onion cells. Again, it's a little bit always a trial and error question, but the staining technique I discovered many years ago just by accident, really. And it actually, if you get the timing correct, it actually works surprisingly well. Yeah. So basically, it's the staining of an onion cell nucleus, and we're going to be starting again. Yeah, I'm just going to show you the desk view again here. We'll be starting again with one of those onion skins. Okay, let's see if I've got one here. Yeah, here we go. Okay, so that's the one. You definitely need tweezers. Without tweezers, it's going to be really a difficult thing. And what we now do first is we need to take water away. I've tried it with salt water, right? Like we've seen now. But another possibility that I would like to show you, and I think I got slightly better results, it would be alcohol. So what I'm going to do now is I'm going to take a little dish here, and I'm going to be adding a little bit of just regular alcohol. For those of you who have been watching this stream and my videos, I think alcohol is a super versatile thing. And look what I'm doing now is I'm just going to dip it into the alcohol, yeah, and leave it in here for, I don't know, a couple of seconds, and it's actually going to turn harder. It's stiffer, yeah. And what the alcohol does is the alcohol, a little bit like the salt water, removes the water, and basically, if you remove the water from the onion cells, then the stain, the ink, is able to enter the cells. Okay, so that's basically step number one. And step number two would be now to I'm going to put the onions away here. Step number two would be now adding of ink. And again, this is sometimes I found this to work extremely well, sometimes not so well at all. I guess it also depends a little bit maybe on the age of the onion, whatever, right? So, and then what you do is this one is just regular fountain pen ink. If you are in a German-speaking country, this is the Pelikan ink, which is essentially the ink that also school children use when they actually start to write. You can try any other water-based ink. I think should also work. Okay. Yeah, I hear that there are some questions here. Just a second. Thank you. I guess I answered again many of those questions already. Yeah, but yeah, we're gonna see. So, and then what I do is I directly add a little bit of how do you say, ink directly to this here, and essentially I just wait, and you're going to see it becomes soft very quickly, and it starts to roll up. Okay, that is very typical, right? And that's why you actually want to always keep it, hold it with a tweezer. Because otherwise, if you have it in a drop of ink, it's going to roll up. It's going to be very difficult actually to find it again. Yeah. So, and then what we're going to do now is I need some water, and I'm going to need this water for washing. So I'm just going to add a little bit more water here. Okay, so that is, So, first alcohol, then ink, now washing. Okay, now it's going to be still too dark. Okay, and now we need to destain it very briefly. So this means that we're going to put it back into the alcohol. Okay. Back into the alcohol briefly, and then back into water. And I hope that this works. Okay, I really hope. We're gonna see. This is basically the principle, and now the most difficult part of the whole undertaking here, of the whole preparation is, and that really takes some patience, how are you going to get it flat? That's the thing really, where you have to be super patient, because getting this thing flat is difficult. Maybe I don't know, maybe kind of trying to fold it apart with your finger is I don't know. Yeah. But that is always the most, you know what I'm gonna try? I'm just gonna try it like this. Maybe like this, and kind of press it on here and then kind of, yep, almost completely flat. Yeah. You see that there is this part here which is folded over. Who cares? This is should be still a single cell, yeah, layer in here. So let's put a cover glass again on top, and again a little bit of water. I'm just going to use this same water over here. And we're going to see if, I don't know, let's see how well it worked. Okay. Ideally, the nuclei should be stained more blue than the rest of the cell. I have no idea. Some of the experiments that I've done like this really worked well. Others didn't. I don't know. It's a question of trial and error, I guess. Yeah. So this is too bright. So let's turn it down a little bit. Yeah. And now... well, actually, I think it did work. It actually did work. So look, now you can actually see that the nuclei are much darker. I think that's, yeah, than before. Yeah. Yeah. So that's basically, look. Here now it's yeah, stained blue, stained blue, stained blue, right? Now, of course, because of the alcohol preparation, yeah, the cells have been killed off. Yeah, so there should not be any movement anymore. But I think it shows quite nicely, and I think the way that the staining worked, is that. Essentially because there are two cell membranes around the nucleus that once the ink has entered the nucleus, either it has stained the DNA and or it was difficult for it to be removed again by the washing process. Yeah. So that's essentially, so I would say that this actually worked quite nicely, and this shows a little bit, yeah, how with a simple technique you can make to do some selective staining of cell organelles. Now there's an interesting question here. What's this silly crown next to our names? And yeah, I honestly have no idea. Okay. Yeah, I know this YouTube keeps on changing. So it says here on top fans. That's interesting. A crown is a top fan. Interesting. So maybe like what happens if I click on it? Oh, okay, I get it. If you click on the top fans, then you can actually maybe these are the number of people who've posted a lot will get this little crown. Maybe, I don't know. Yeah, so this could be a new feature. Maybe it's a new feature of YouTube. Yeah, that those people who kind of post many questions will actually receive a nice little crown. Yeah. Yeah, just wanted to show this to you, as a little staining experiment. Okay, and what I'm going to do now is I would like to switch topics, right? And I would like to quickly show you how the hydrogen peroxide treatment went, because here I got a really unexpected result. Yeah, actually not a result that I was hoping for. But I would say probably more interesting. Yeah, Marie is asking, decides who? Well, YouTube's algorithm. YouTube's algorithm simply counts apparently how many times somebody's posted, and maybe will automatically add those little crowns or logos next to the name. I assume. Yeah. So you see, YouTube is always trying out new features. Okay. Yeah, so just that's kind of the thing that I just wanted to show you, right? And yeah, just keep the destaining time short, otherwise the blue nuclei are also gonna start losing the color. So yeah, back to last week's little experiment. Last week I added, if you remember, hydrogen peroxide to this here, and it started to bubble a lot and formed oxygen. And today I checked it again and actually found a little white deposit right here on the bottom. And I said, wow, cool, maybe you can, I don't know if you can see it here, there's some white stuff here. I said, wow, cool, these are diatom shells that have separated. And actually, yeah, some of them. Something that I did not fully expect, and I'm going to show this to you. And of course, every time, I just need to say the following. Normally, if you basically get the result that you expect, you're not making a lot of scientific progress. You got to get results which are different, because then you actually might have, you've learned something new, right? And same here, because I actually have to tell you, I was a little bit surprised by what I was able to find. So I'm going to take a little sample here, and I was hoping for some kind of diatom shells or whatever, you know. So, and I'm going to put this down here because, for those of you who were not here last week, what I've done is these are some water plants from an aquarium, some algae actually, full of diatoms, and I added quite a concentrated 30% hydrogen peroxide, which you got to be careful with. And it started to bubble a lot, and it started to break down the organic matter. And usually this causes the diatoms to separate and fall down. Essentially then you have a more or less pure collection of diatom shells. And this was kind of what I was hoping for. And what did I get? Yeah, I hope that now I'm able to show you the same thing that I was able to find before. Cover glass goes on top. Okay, I'm going to remove the old slide. I'm going to add the new one. Yeah. So, and let's have a look. I need to find the focus again, of course. Okay, and you know what? I'm gonna switch over to DIC a little bit, because it does look a little bit nicer. And this is what I got. Those square things that you see could be indeed some diatom shells here, and those tiny diatoms here. But there's something that I don't understand. Just looking at this. Let's go up here, and you've already seen it before. This is something I do not understand. What did I add? 30% hydrogen peroxide. What do I see here? How in the world were they able to survive? That's the question I have. I mean, 30% hydrogen peroxide should kill off everything. I mean, you're using 3% hydrogen peroxide for disinfection. And look what I'm getting here. Am I going crazy here? Yeah, all those tiny dots in the background that you see moving around, bacteria. How in the world did they survive? Okay, I mean, I have a hypothesis, okay? I have a hypothesis, but this was actually, of all of the things, this was unexpected, right? Yeah, of course, there are some diatoms here, but not as many diatom shells as I expected, right? But they really look, okay, that's a nice sample for phase contrast, of course. Yeah, bacteria. I mean, look at this here. Look at this movement. I mean, that's phase contrast now, right? I mean, this thing that's crazy. I mean, I added 30%. You know what happens if we add 30% hydrogen peroxide on your skin? I mean, your skin's gonna start peeling off. Yeah. So you're asking spores and cysts, possibly. But look, even, I don't know, some ciliates or flagellates, or even larger ones are floating around. So what I suppose happened, and I'm guessing now, and maybe sometimes it's multiple reasons can play a role, is what when you look at the sample here, you can see that it is not fully digested, right? It's still green and everything. So essentially what I assume happened is that the hydrogen peroxide, essentially, of course, killed off many organisms in here, but it was used up in the process, making oxygen, because many bacteria and microorganisms have an enzyme called catalase, and this catalase breaks down the hydrogen peroxide. Yeah, that's because those cells want to protect themselves from hydrogen peroxide. So I can imagine that even though the hydrogen peroxide was so concentrated that essentially it was broken down faster than it was able to kill the organisms, right? And then maybe a few of them did survive, and then they kind of kept on reproducing. And because hydrogen peroxide is broken down to oxygen and water, basically there a lot of oxygen was also present. Right, so this basically means it's probably an ideal. At the beginning, I killed off maybe a lot of organisms, but then later on, basically, yeah, plenty of oxygen was present, and then all of those other microorganisms could reproduce like crazy. Yeah, so that's, or it was as commented, maybe it was not distributed evenly. That's also a possibility, right? That certain parts maybe could not, the hydrogen peroxide could maybe not reach some of the parts. Yeah. So generally, what I probably should have done is I should have after it stopped bubbling, I should have allowed all diatoms and whatever to settle, and then remove the old hydrogen peroxide and add new ones, and keep on doing this until all of the water plants are digested and removed. Right. I of course didn't do that. And so this kind of shows a little bit, that was a little bit surprising for me, how can they survive, right? Evidently they did, and the hydrogen peroxide was strong. We could see the oxygen bubbles. So for those of you who were not, just check last week's live stream. Evidently a lot of microbes survived. Yeah. So in that sense, I don't know if I should consider this experiment a failed experiment or not. I wanted to concentrate the diatoms, but actually I discovered something more interesting here. Yeah. So what if we add isopropyl alcohol first to kill everything, and then pour it off, and then add hydrogen peroxide? Yes, maybe possible. I thought that hydrogen peroxide should already be a strong enough disinfectant in the first place. This was kind of my assumption, right? So because it's super aggressive, right? So, but again, that's one of these things where we discover new things here, right? So I'm going to go through some of your comments here. Okay, so maybe the hydrogen peroxide has degraded. Well, it certainly has degraded after I've added it, because it made the oxygen bubble. So it was, of course, a degrading process by the catalase enzyme, definitely. But when I added it, it was still fine, because it made the oxygen bubbles, right? We could see that. Yeah. So spores and cysts, possibly. Yeah. Hydrogen peroxide does break down when exposed to air. How old was your bottle of hydrogen peroxide? It was already a little bit old, right? But that it still worked. We could actually see last week, because it still created those oxygen bubbles, right? So it was able to actually break down. Yeah? So there must have been something left. So maybe not original concentration of 30%, maybe somewhat less. Possible, but evidently still enough. Yeah. So, or if the solution was too diluted, or some film happened in the protected life at the bottom, or some bacteria resisted. I mean, of course, all this is possible, but I mean, hydrogen peroxide is basically oxidizing pretty much everything that's organic. Yeah. So bacterial resistance. I mean, never say never in biology. Nature is so diverse, right? Would also be, I mean, if there are some bacteria that were able to resist hydrogen peroxide of that concentration, I mean, that would also be quite an interesting discovery. Yeah. Okay. Yeah. Also already mentioned that maybe it was not evenly distributed and so on. Yeah. Yeah. So what I'm going to do now, very quickly, I'm going to comment on some of those, and then I'm going to see how many questions essentially are on the sheet here. Okay. I use Helicon Focus software to focus tag images. So do I. This can take a few minutes. The problem is invariably subject matter on the slide moves and the final result image is useless. That is generally a problem. So if you want to use image stacking on moving objects, I think the stacker has a problem, right? And you should actually use it and only on permanently mounted slides. Yeah. It worked. Maybe it was weaker than expected. I probably think so as well. Yeah. So because indeed, so this is actually one of the surprising things. What do you do in the sciences when you discover something weird? You repeat the experiment. Yeah, that's the first thing that you want to do. Yeah. So yeah, you repeat the experiment and you see if you're able to regenerate that what you've observed before. Yeah. So what I'm going to do now is I'm going to switch over a little bit, but again, I think I might have already answered many of those questions already. Yeah, but just yeah, let me quickly have a look. Let's the desk view. Yeah, because sometimes yeah, there's some questions here that I might have overlooked. Maybe in a future stream, extract the pigment. Okay, so that's the next thing that I might have to try. Is this pigment actually, is this pigment over here actually alcohol soluble or not? Yeah, so yeah, that's something that you know what? 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 I've got some alcohol here from before, remember? Yeah. So let's, why not just drop it in here and see if, yeah, if, yeah, I mean, of course, the alcohol is not able to reach all of the plate parts, but maybe, oh, wrong way around. But maybe, yeah, if we see that a little bit of the red pigment comes out from the side, maybe it is soluble in alcohol. Yeah, it does already turn pink a little bit. Yeah. That would be actually, if alcohol, maybe I have to, yeah, crush it a little bit more, or maybe, you know what, I could try, is this, look, this one over here, maybe. Let's try it. And because if the red pigment indeed does dissolve in alcohol, by evaporating the alcohol, you can actually make it more concentrated. And look, it does seem to turn pinkish a little bit. Yeah. So we're going to leave it a few minutes here. Can we try to stain organelles someday? Yeah, difficult. You need kind of specialized stains for that. Okay. But at least the nucleus of the onions, I try to, that's a cell organelle, I'll try to stain that with you. Yeah. Okay, the areas without the dots, these are the vesicles in the onion. Okay. I already mentioned that, basically that in the center of the plant cell you have the central vacuole and all the other cell organelles compressed to the outside. Okay, so maybe at some point to do contrast staining or differential staining with 2 or 3 different stains on a single sample to highlight different parts. Would be cool to see if we can do this. Yeah, it should be possible. Not tried it out so much yet, but certainly something. So basically using different stains. You do this actually with plant tissue, so that plant cross sections, you can use different stains to stain the different parts of the woody parts of the plant stains differently than the non-wood parts and so on. Yeah. So this is actually done. However, I don't have all of those stains yet. Okay. We talked already about the laser diodes. Okay. And what I just say is yeah, I don't know if you're using a laser, because it's a very parallel light, yeah, to what extent it might not cast very strong shadows, therefore. Maybe sometimes it'll, yeah, one would have to try out. Yeah. Okay, so that's essentially using some kind of laser diodes for illuminating a specimen from the side. Yeah. Okay. Yeah, the outside edges, I already mentioned that, right? That has to do with the way that the cell, yeah, the plant cell at least is structured. Different light. Ultraviolet light is fluorescent. Infrared, I don't know so much about infrared, if this is used. Generally, the problem with infrared light is because it's long wave. For this reason, it has a lower resolution. And it's actually also thermal, so I don't know if, yeah. It would be kind of interesting, actually, on the other way around. I mean, this is, I really don't know anything about this, but, you know, living things do, even plants, because of chemical reactions happen, even plants generate some heat. Right? It's kind of unusual because simply of chemical reactions happening. And I wonder if, and if a little bit, tiny bit of heat is generated, then infrared light is emitted. So I wonder if maybe you have some kind of camera systems out there which are able to do infrared photography of microscopic images to see which parts of an organism or which parts of the cells are metabolically more active, because these parts maybe generate a little bit more infrared light. I'm brainstorming, yeah, but that would be kind of interesting, yeah. This is the kind of channel where I saw this 10 years ago. I would have majored in microbiology. Oh, thank you. Okay, I need to thank you very much. I feel very honored. Now comes the big but. The big but is that unfortunately, when you study microbiology or molecular biology, you don't work a lot with microscopes, especially in molecular biology. It's very biochemical, and generally microscopes occasionally are used, very often fluorescent microscopy, but depending on the research area. But I was a little bit disappointed when I studied microbiology a long time ago that microscopes were not used more often. It depends a lot on the research area, but microscopes are used for quality control and for characterizing microbes, but at least in bacteriology, where I was working in, unfortunately, microscopes were not frequently used. So if you're interested in going into the biology topic, always make sure it depends very much on the specific area of research and the research question that you're actually investigating on whether you're going to be using microscopes or not. Okay? The elasticity of the cell membrane I talked about already. Okay? I talked about the tap water. What concentration of salt water did you use for this experiment? Honestly, it's concentrated. It doesn't matter as long as it's sufficiently concentrated. I just added salt and added water, and basically it's saturated salt water. If no more salt dissolves, then it's maximum salt concentration, but it's not even necessary. Yeah, it's not even necessary. Yeah. But I didn't even weigh anything or did not do any concentration calculations or anything. Yeah. Can we see the crystals at the end of the session? Which crystals? You mean the salt crystals? Okay. Oh, the salt crystals. The salt crystals. You know what I'm gonna do? Yeah, we can do that. I'm gonna drop a little bit of salt water on the slide. Huh? How does the pigment concentration show in phase contrast? Ooh, I didn't do that yet. Do we have this? I don't know where the slide is. Yeah, I don't think that the pigment will actually show much. Yeah, I think I already discarded the slide a little bit. Okay, but in phase contrast you would not see a lot. Okay, that's the thing. Yeah, are you getting a greater concentration of diatoms? Yeah, we just talked about this. And if you wash the sample in water and then put it back on the slide, would the rehydration happen faster? I also talked about this. Yes, it probably would happen faster, but you would not be able to observe it under the microscope. Okay. Can you put a drop of hydrogen peroxide? Can we see the pigment change color due to the pH change? The thing is, I mean, I could try to do that, but I don't think that the hydrogen peroxide will change the pH too much, honestly. But the question is here is the one, here's the sample, but it already lost all of the color. So the question is, I guess, put a drop of hydrogen peroxide and see. You know what, we could try. I did not try that. Let's do another one here, and let's simply add hydrogen peroxide and see what happens. Okay? Yeah, I have no idea. Never tried this, and that's why we're doing it. So, let me cut another one off here. Yeah, and here we go. Just gonna take this one over here. Oh yeah, onion. So we go here. It's a little bit out of focus because. But you get the idea. Here we press down again and we carefully rip it off. Here we go. Okay, this worked nicely. I'm gonna put it on here again, flat. And now we can actually see how it looks in phase contrast and also when we're adding hydrogen peroxide. I'm going to use again my how do you say this, knife here. So here we go. We want to remove this thick, bulky part, spread the rest flat. Okay. And let's put this away. I'm going to add another cover glass, where it is. Let's press it flat. So, I'm just going to, so that's basically where we were before. So So this is now, let's just have a look, a regular look on bright field and how it looks in phase contrast. Let's add a little bit of hydrogen peroxide and maybe nothing's gonna happen. We're gonna see. Yeah, here we, I still have to, so this is the, let's go up here. This is just regular bright field. So that's, these are just the normal, these are just the normal onion cells again. Here, now they look a little bit different in shape, but actually it's all kind of nice. Look, where is this? I have to close the condenser. See, the here it's kind of funny. The nucleus, it's nice. The nucleus looks brighter, and everything else is pigmented. Okay, so that's basically these are the red onion cells. Actually, it's a pretty nice specimen. Again, so let's try it in phase contrast. Why? Because you wanted to see it, and I have not seen it myself. And we have to turn up the light. And this is how it looks in phase contrast. Oh yeah, just the cell membrane, cell wall looks much brighter. Yeah. Look. But do I see some movement in here? Maybe we're, yeah. Look, I don't know if you're able to see this now. I mean, those, some of those dots are kind of bouncing around in here. That could be Brownian motion, right? But sometimes what you're able to see is also that some of them are kind of moving in a directional manner, and then these are vesicles that are being pulled around. Yeah. Yeah. So let's do the following. Let's add a little bit of hydrogen peroxide. And see what happens. I can imagine that some oxygen bubbles might form. Yeah. So, but I'm going to go down now. This here I have to be really careful, folks, because I really don't want the hydrogen peroxide, which is very aggressive and caustic, to touch any parts of the microscope. Yeah. So this is regular bright field again. I'm just quickly gonna get my H2O2 bottle. Just a second. If I can find it. I've got so many bottles here that I have problems finding it. Just a second. Gee, I can't believe it. Where did I put it? Just a second. Ah, it's over there. It's I put it next to my window. Sorry about that. Okay, so that is I've got a little bit of hydrogen peroxide left. So let's. I'm gonna pour in a little bit into the desk view. So I'm gonna just look, not more than a drop, please. I don't know. Maybe you're hoping for super interesting things to happen. Maybe nothing's gonna happen. Okay, so please don't be disappointed if nothing happens. Okay, just saying. Just saying. Okay. So let's a little bit of hydrogen peroxide here. You do not want to get this into your eyes, obviously, right? And where is the scope view? And then after this here, I think I'm just gonna call it quits in a day because I feel really hot already. So I'm gonna go down with the magnification because I want to I want to make sure that there's a safe distance between. And now let's add some of this hydrogen peroxide and let's see what happens. You see that it kind of was spreading a little bit. And let's see if there is a possible color change or not. Maybe add a little more. Okay, now it seemed to have covered it. And what happens? Nothing. You can actually see how it's spreading. Yeah, so correct. Ever small is saying nothing, but might need to break the cell wall. Could be. Yeah. On the other hand, actually, the cell wall should allow it to go through. And then maybe what could be the case is that indeed bubbles, that as it breaks down, it forms oxygen. So maybe some of those bubbles will expand as the hydrogen peroxide breaks down and forms oxygen bubbles. Yeah, and they do seem to be growing a little bit indeed. Basically, what you can see every now and then is if you add hydrogen peroxide to water microorganisms, that essentially they will be killed, obviously, but this is also the place where hydrogen peroxide bubbles, oxygen bubbles are forming. Yeah? Now, see, those bubbles actually are growing. So the growing bubbles are actually an indicator that the hydrogen peroxide is breaking down. Yeah. But no color change. So just a second. Now I'm a little bit curious. Let me try something else. Just a second. Yeah, I'll be right back. I'll try to get some acid. So I'm back. I got some. This is 20% acid, acetic acid. Because you see, hydrogen peroxide does not change the pH so much, but obviously acid does. So what I'm going to do now, I'm just going to remove this. I'm just going to remove this, and where's the desk view again here? Yeah. So this is I gotta be careful with this thing now, right? And here I gotta be careful. Maybe I don't know if you see there's some tiny bubbles here on the bottom. I'm worried about this. So look, what I'm doing, I'm diluting it with a lot of water, simply for safety reasons. Okay. Yeah, that's it. Yeah. So let's put it over here, and let's give it another try. And so this is basically where I added some of this acid here, right? So what does it say? 20% or something like that? I don't know, 25. So it's way stronger than regular vinegar, and it's used for cleaning purposes. Also, you can actually if you need to put it on your, I don't know, salad dressing. Yeah. And this is actually quite interesting. I've didn't think of the possibility of actually doing a pH, using this as a pH indicator. But I don't have a base. So same. This is a little smaller. This is a little bit smaller. Again, this bulky part I have to try to cut away. Yeah, this is gonna be fine. We're gonna put where's the cover glass on top? Yep, and let's have a look. is the scope view. It takes again, I think, too much time for it to appear. Here we go. Yeah. Okay, here maybe that's a. Again, you see, yeah, here you see that the cells that are ripped open on the edges here are white. So it doesn't matter. We're not concerned so much about that. So let's have a look now. And now I'm going to, where is this here? This is a fresh tip. I don't remember. Let's use a fresh tip. And let's add some of this not highly, but somewhat concentrated acid. And let's see if there is now a color change or not. I don't know if it reached the yet or not. Otherwise we're going to add a little bit more. Let capillary action do the rest. It's a little too far away, I see. Now it touched it. I need to refocus. Ooh. I don't know if there's a color change from the outside. Capillary action, of course, yes. Pulled the liquid beneath the cover glass. But I don't see a huge difference in color. The question is, it turned a richer pink. That could be. Yeah, one would have, I would probably have to try maybe also an alkaline solution, which I don't have now. But at least that's, what should I say? Yeah. At least then we have another thing to investigate. Because if that would have worked, it would have been actually quite nice to actually see how those cells respond to pH. I just skipped back in the video. It does look a little bit richer. Okay. I wonder if this could be also simply because of a liquid being present, so one would have to do a control. I would say I somehow have to organize myself a stronger base, alkaline, and then see if this is actually makes a difference. Yeah. Let's have a look very briefly here. Yeah, because this was the question from before. I'm going to switch over back to the desk view, and then I think I'm gonna call it a day. Look. Yeah, it did actually was able to leach out. Look, let's use a white color. Yeah, it was actually, the alcohol was actually able to get out a little bit of the pigment here as well. Yeah, so look what happens if we hold it like you. So actually, maybe it does work that alcohol is able to remove the pigment, and in this case, by evaporating the alcohol, we should be also be able to get a stronger pigment. Yeah. So I guess. Yeah. Yeah. So you see, I'm playing around a little bit, doing some kind of experiments here, and yeah. But I think what I'm going to do for today, where is the microscope again? I don't know. Something is, I wonder if my USB system is acting up again because it's not switching cameras quite as quickly. Yeah, but I mean, that's basically how it looks like now. Yeah. Yeah. So what I'm going to, look, it's interesting look how it makes this smooth transition actually. From those cells here. It's also quite nice. Interesting. Huh? It somehow appears a little bit like, I don't know, before the cells were kind of separate. Before that, some of the cells were white, others were, and now kind of everything is kind of much smoother. It's like a gradient. Huh. Yeah. Okay. You know what, folks? I think I'm just gonna leave it as it is now. Yeah. I'm going to call it a day. I've been already talking anyway, almost 1 hour and 40 minutes. So it's all the time for me to go to bed. And I think I'm just going to call it a day. Thank you that you joined me again today. Do leave your comments. You probably have, on an organizational note, you probably have noticed that in the last couple of weeks, I did not post as many regular videos as I used to do. I have to be honest. It's the end of the school year now, and it was extremely stressful. But now it's getting better a little bit. So I hope that in the next upcoming weeks I'm able to generally do a little bit more videos and yeah, in this channel here. I'm going to call it a day today. I wish you all the best and happy microbe hunting as always. Hope to see you again next week. And if for whatever reason it doesn't work out, please do check the trailer or the announcement because yeah, it will always have this countdown and it will tell you when the live stream starts. And if for whatever unexpected reason I'm not able to do it at that time, then you'll know in advance as well. Okay, all the best. Bye bye. Have a nice week.